This Week in Virology, the podcast about viruses, the kind that make you sick.
From MicrobeTV, this is TWiV, This Week in Virology, episode 1273, recorded on November 21st, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here in Ann Arbor, it's 51 Fahrenheit, which is 10 Celsius. White, puffy clouds, blue sky, and a light wind.
Chelsea, New York, it's 11 C and cloudy. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see you all. In Madison, New Jersey, it is also 51 Fahrenheit, 10 C, and also cloudy.
We have got great papers for you today, but first, if you enjoy these paper discussions, you can learn from them. They're sometimes fun also. Well, for us, it's a lot of fun. Maybe for you too. We'd love your support. Go to microbe.tv/contribute. Why do we want your support? Because we don't do ads to pay for these programs, and so we depend on you. I decided a long time ago I didn't want ads. Okay, there's a great cartoon in the New Yorker. I have to get it. Hold on. This is so funny. It's entitled, "What Microphone Should You Use?" And there are 4 different kinds of microphones, right? A guide. First, there's a headset with a boom, right? And it says, "You are a self-aggrandizing lecturer in a turtleneck or working in an overseas call center." And I resent that because I'm a lecturer in a turtleneck, but I'm not self-aggrandizing. I feel a little guilty.
When I wear one kind of like that in class, when I used to wear one that was more headset-y, I may have been called Britney Spears.
May have been.
Then they have a Shure SM58, which the rock stars use, and say you're screaming over a snare drum. Okay. Then they have a fancy mic like this, and it says you are babbling endlessly amid Casper mattress ads, and that's what, right? All the podcasters do ads for Casper mattresses. And then there's another kind of mic. You are a snare drum, which is a mic you would use for a snare drum. But I like babbling endlessly. Is that what we do? Do we babble endlessly? No, only 2 hours.
That's not endless.
There are no Casper mattress ads here.
And we don't do any ads because otherwise we'd get made fun of in a New Yorker cartoon. So please help us, microbe.tv/contribute. Remember, Immune 100 live at the incubator. It's coming up in December 22nd. Starting at 6 p.m. right here in the incubator. So, if, we figure local people would want to come. There's not a lot of space here, maybe 20, 25 seats. And you need to go and buy a ticket, which would be a donation. And there's a link at microbe.tv right at the top of the page there if you're interested. And we'll have Brianne and Steph and Cindy and myself. And then we have 2 local guests. So should be fun. And then one news item, which has dominated the news since yesterday. I picked an NPR article, but there are dozens. The CDC revives debunked, quote, link, unquote, between childhood vaccines and autism. So if you go to the CDC website about vaccines, it says autism cannot be ruled out. The link between vaccines and autism now can no longer be ruled out. Because they say you cannot prove that vaccines don't cause autism. It has never been proven. And in fact, as Paul Offit says, you couldn't prove that chicken nuggets don't cause autism. You can't prove that anything doesn't cause something.
Right. The one on NPR this morning was something about that umbrellas don't cause, and it wasn't even autism. It was umbrellas cause something. Yeah.
Umbrellas don't cause rain, right?
Well, no. And then Brianne and I use the ice cream cones in summertime, and
Ice cream and drowning.
And
Yep.
Yeah.
And this article goes on to point out that so many high-quality studies have shown no association between vaccines and autism. And so that's the best you can do with an observational study. And because multiple studies have come to the same conclusion, that we say there's no link, and so it is safe to have vaccines. Autism has other causes. But now, as you know, RFK Jr. is directing what's on the CDC website, and so he is an anti-vaccine person and wants to sow more doubt into people's minds. And so this is dangerous because it will cause more people not to get their kids vaccinated, and there will be deaths and disease.
This is the quote that I was looking for, and I meant to go back this morning and look at it, but you have it right here for us. The president of the American Academy of Pediatrics said, since 1998, independent researchers across 7 countries have conducted more than 40 high-quality studies involving over 5.6 million people. The conclusion is clear and unambiguous. There is no link between vaccines and autism. So, here we're kind of preaching to the choir, but if you want to try and remember those statistics, 40 high-quality studies, 5.6 million people in 7 countries.
Yes.
And notice they say there is no link. That's the proper way to say it.
That is the proper way to say it. And having just done a whole class period on this the day before they made the change, I can remind you that the study that people often cite on the other side, so we've got 40 studies with 5.6 million people, kind of versus one study with 12 people.
Yeah.
Right. Are you going to go back to your class and say, look what happened since we last met?
I did already, yeah.
Oops.
Yeah.
Okay, good. There's a quote here from a CDC scientist said, the scientists did not participate in this creation and the data are unvetted.
Yeah. I was not totally calm. You said it in a much calmer way than I said it to my class.
Show us how you said it.
I don't know if I'm not sure if I can do it sitting down.
Okay.
It's hard to wave my arms around enough.
Were you wearing a turtleneck?
I don't think so.
Do you think you're self-aggrandizing? I know you, and I don't think neither of you are self-aggrandizing. But I've been accused of being self-aggrandizing, so that's why that kind of bothered me, that cartoon. I don't think I am. I really like to teach people, but, what are you going to do? Okay, so a few weeks ago, James Watson died. We talked about that on TWiV 2 weeks ago, and so I thought we would do the 1953 paper, A Structure for Deoxyribose Nucleic Acid. And in the same issue of Nature, there were 3 papers on structures of DNA, and this is the first of the 3. So they're all collectively called Molecular Structure of Nucleic Acids, and so this is the first one, A Structure for Deoxyribose Nucleic Acid. So the authors on this, of course, are James Watson and Francis Crick, who at the time were at Cambridge University in the UK. And there are no experiments in this paper. There's no computational biology. There's a little bit of model building and biochemistry. And I was unable to highlight this manuscript for whatever reason. All the other ones I can highlight, but it's short enough so that it doesn't matter. The first sentence, we wish to suggest a structure for the salt of deoxyribose nucleic acid, D period, N period, A period, which of course nobody uses the periods anymore. This structure has novel features which are of considerable biological interest. And I'll link to an interesting commentary article which says that this paper is written really well because the other 2 papers begin with completely impenetrable. Here's the other one of the others. While the biological properties of deoxypentose nucleic acid suggest a molecular structure containing great complexity, X-ray diffraction studies described here show the basic molecular configuration has great simplicity. It doesn't really, it's not as straightforward. Right?
No, I agree. And I kind of forced myself to read the other 2 yesterday.
Yeah,
and
too.
I had to force myself because it's true. The Watson and Crick paper is simple, clear, to the point, and accessible.
Mm-hmm.
And the other 2 are written for a very narrow, high functioning audience.
Well,
yes, for sure.
I also am as I'm looking at this, very amused by the next sentence or the next couple of sentences where they reference a previous structure from Pauling and Corey that they have looked at. And which they say is unsatisfactory.
Right.
I feel like that's quite the understatement.
Well, it's a triple helix, right?
Yeah, it's a really, really not good structure.
And so it's Linus Pauling. You would think and they call him the greatest chemist of the time, but they made a triple helix with the bases on the outside, and there was already evidence that wasn't the case. So they
when Watson and Crick did their model building, they did do a triple helix model for a little while.
Yeah,
they tried
that's
it,
right.
So
it
didn't seem completely out of line at first, I guess.
So what's interesting about this paper is actually the backstory. So let's just present the results first, and then we'll tell you the backstory, which is cool. Then there was a second 3-chain structure, which was actually never published by Fraser. And in that one, they put the phosphates on the outside, but it was still 3 strands. And they say this structure is rather ill-defined and we will not comment on. Okay. So these guys are really snarky from the beginning. So they say we're going to put forth a radically different structure, 2 helical change coiled around the same axis. And they're making assumptions that each chain has 3 to 5 prime linkages in it. And they're right-handed, and they go on to talk about helices, and they go that there is a residue on each chain every 3.4 angstroms, which is correct. And they got the angles and so forth, and they say it's an open structure, so the water content is very high, but. Okay, so first of all, it's a helix, but other people had suggested a helix already. That's not novel, and we'll see the other 2 papers. We'll go over that briefly. But the novel feature here is the way they're held together by the bases, the purine and the pyrimidine bases, right? The planes of the bases are perpendicular to the axis of the fiber, and they're joined in pairs. One base is on one strand joined to another base on the other strand. And you always have to have a purine and a pyrimidine for binding to occur. And they made some models which reinforce that. And I'll tell you how they came upon that in a bit. And they say, if you assume that the bases only occur in the most plausible tautomeric form rather than the keto, rather than the enol configuration, then you can make these pairings. And the pairs are A, T, and G, C, right? And they say that this now follows Chargaff's rules, which said that there's an equality of the A and T and G and C. The ratio of those are always equal. So,
can I insert something here? Because in
Yeah,
one
of
of the
course.
things I read, it says that because they were doing this model building, and when they got this hint about looking at, which did you say, the keto instead of the enol?
The keto instead of enol.
Right. That when they did that, and they had to make new cardboard models of the bases, that in rearranging them, that's when they saw that the AT one paired together kind of came out to be the same size as the GC.
That's right.
So that was kind of cool.
That's so you could accommodate it between a fixed helix. Yeah,
right.
That's actually it.
Right.
And so Chargaff had found that the ratio of AT and GC were the same in all nucleic acids. Chargaff's rules, it was called, but nobody knew why, and this explains it completely. And so that's really it. And then they say it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And that's it. And so full details of the structure will be published elsewhere. I don't think they ever were. I think this is really it. There's one picture here, the double helix, which apparently was drawn by Mrs. Crick. I forgot her name.
Odile?
Odile, yes. It's a very nice picture.
It is. It is a frequently seen picture in biology.
Yeah, it was even in the Mrs. and Stahl paper that we did.
Yeah.
I think they absconded this picture. So that's the paper, and there are 2 more behind it, as I said. But I just want to tell you a little bit about the backstory. So initially, Maurice Wilkins and Rosalind Franklin started working together on the DNA structure. And this is when Franklin produced very nice X-ray pictures of crystals of DNA, which showed that it was clearly helical. But apparently the two of them couldn't get along, so they split up and continued working independently. So the two papers that follow Watson and Crick, Wilkins is on one and Franklin is on the other. Now, then the way it worked is that Watson apparently visited Wilkins one day, who was at another institution, and Wilkins shows him this X-ray, this famous number 51, and Watson says, "My God, it's a helix. There's no question." And he went back and they worked on the idea that it was a double helix, okay? So kind of took, and Wilkins had never asked Rosalind Franklin for permission to share that, so that's kind of snarky. But anyway, why are we even looking at the structure of DNA, right? You know, in the 50s, we used the word gene, but nobody knew what a gene was. Now, in 1944, Avery, McCarty, and McLeod had showed that DNA can transfer features, phenotypes among bacteria. So we knew DNA was the genetic material, but no one knew what DNA looked like.
Right,
so,
because there had to be experiments to show that it wasn't protein also.
Yes.
right. So that's why Watson and Crick and others are working on the structure of DNA, because you need to understand the structure to know how it works. And apparently, so other people had been working on this. So an organic chemist, Alexander Todd, had determined that the backbone had repeating phosphate and deoxyribose sugar groups. Erwin Chargaff found this equivalency principle that we talked about. And Wilkins and Franklin showed that there was a helical shape to the molecule. And then, you know, Pauling was working on this at the same time. Pauling, of course, had discovered the alpha helix in proteins. And so he also did a lot of model building, which is what Watson and Crick used. And they actually thought they were going to get scooped by Pauling, but he proposed this triple-stranded helix, which was wrong. Okay, so as I think Kathy mentioned, they had some failed attempts even with their three-stranded. And that's when this chemist from the U.S., Jerry Donahue, comes in and he says that the configurations of the bases in textbooks of chemistry were wrong, right? So they make little cardboard cutouts with the right orientation, and Watson is playing with them. And suddenly he realizes that when he pairs A and T and G and C, they're the same width and that they could exist between double helices. And it also then explains Chargaff's rules as well. So that's basically how they did it. They used the X-ray structure of others who showed it was helical, and then they made this interesting chemical deduction. Now, the other two papers, so the one immediately next to it is molecular structure of deoxypentose nucleic acids. Instead of saying DNA, right, they're using a more chemical. And there's a X-ray image. And the authors on that are Wilkins and Stokes and Wilson. So this is Wilkins after he split off from Franklin. And they basically conclude that it is a helix, right? And they do a lot of very obscure chemical stuff here. They don't say anything about the bases. Basically, they're saying it's a helix. And then the third paper is molecular configuration in sodium thymonucleate, which is basically DNA, the sodium salt of DNA from calf thymus, you know. And this is Rosalind Franklin and mine is cut. Oh, Rosalind Franklin.
Is it like Gosling?
Gosling.
Yeah, something like that, yeah. And there is the picture 51, I think.
Yeah,
very nice
photograph 51.
X-ray showing the helical structure. And they say the X-ray diagram shows in a striking manner the features characteristic of helical structures. So that's what the two papers. So those two papers concluded it was a helix based on data. And so Watson and Crick just took that and ran with it. Yeah, Franklin and Gosling at King's College. All right, so why is it it has not escaped our notice? Well, if you've got the pairing of bases, AT and GC, then if you separate the strands, you can make a new second strand by just the enzyme. It sees an A, it puts a T in. It sees a C, it puts a G in. So it's a beautiful idea of how to copy a DNA, which of course was subsequently proven by many other people.
And it also relates to the fact that then you can have those nucleotide bases in any order.
Right.
And that could give you lots and lots of different variations, as we know.
And then, of course, in the subsequent years, people realized that the code was a triplet. They started to decode it, starting with polymers like UUU, right? They could, it's phenylalanine. And they decoded the whole code and figured out, they discovered mRNA and tRNAs and so forth. So this whole molecular biological investigation begins with the structure of the DNA. So I think 53 is the beginning of the era of molecular biology. And it's the year I was born also. So I absorbed all of that. When was this paper published?
53.
What month? I'm going to know what month.
April 25th.
25th.
A couple months after I was born. That's why I wanted to be a scientist, I think. So that it's really a landmark paper because it starts so much else. But they stood on the shoulders of giants, right? They didn't do it all themselves. But there are two individuals sitting around thinking and talking. They're obviously smart and had good ideas. And this is what came of it.
And they wrote well.
So the writing is so good. Yeah, there's a nice article here, which I will link to. It's called 3 Papers, 3 Lessons, actually. And it's open access, which is kind of interesting. The first lesson is that collaborating is more profitable than competing against them, right? So, which is true, but people don't know that. Second is that research has no value if it's not published. And there was, some unpublished stuff that never amounted to anything. And the third lesson is that the contrast comes from the contrasting prose presentations. Watson and Crick have something to say. They say it and shut up. The article exemplifies the 3 qualities of scientific writing: brief, clear, and simple. So that they use half the number of words than in the other papers. And it's very clear that their writing is lovely. Yeah. Whereas there's a lot of mathematics in the other paper and just it's not clear what they're trying to do. All right. Any other comments, thoughts on this paper?
No, but I'd like to talk a little more about the history.
Yes.
Okay.
I'm sorry. That's what I meant.
Yeah. So Rich alluded to some of this when he was talking, about Watson and there's stuff in the obituary as well. But for a couple of reasons, people have different amounts of animosity toward James Watson or Jim Watson or some of the things that he did. And one of the things that he did was write a book called The Double Helix. And it was very popular. And I haven't reread it. I maybe reread it once a long time ago, but I haven't reread it. But it's probably similarly pretty well written and easy to understand. So I think and it's not very long.
Yeah, I mean, we had to read it in college.
Right. So, so I think a lot of the non-molecular biology public has read it. And, but in it, he really does not treat Rosalind Franklin very well. And,
I think even not in the book, but in real life, he didn't treat Rosalind Franklin really well.
Well,
is the impression that one gets.
Yes, and no, because one of the articles said that they had a friendly relationship, and she might even be surprised by all of this stuff, kind of making her out to be the wronged heroine. He offered to drive her to California when he was going, and she was coming over or something, and so. But he calls her Rosie, a nickname she never used. And you can just imagine being the only woman in that situation, that cadre of 4 to 6 to 8 people working on trying to get the structure of DNA. She would probably not get a fair shake. It wasn't that she was the only woman there at King's College or in Cambridge or whatever. There were others, but at least in this kind of thing, that was the case. Mm. And so I don't think that he, in subsequent years, maybe ever righted that wrong of how he wrote about her in the double helix. At least I'm not aware of that. Okay.
I am also not aware of that.
And so
many things I'm aware of point in the other direction.
Right. And so then, Watson and Crick and Wilkins received the Nobel Prize, but it's something that the Nobel Prize Committee didn't even have to deal with as to whether Rosalind Franklin was eligible, because she was not eligible because she had died. And the Nobel Prize is not given posthumously. So that's, life is unfair. And so I don't have any problem with her not getting the Nobel Prize, other than, she had, was it ovarian cancer, right? Mm-hmm. So, yeah. And died at 37 or so. So that's unfair, but that's not a human unfairness. And then, then Watson went on to a great career. He became the head of the Cold Spring Harbor Laboratory. And then I think Rich alluded to this, but I don't know if he said it. But then, Watson really said some pretty outrageous things about genetics and race, and kind of became a genetic determinist. That the whole lot depends on your genes. And in fact, when his son had some kind of mental illness, he just said that his son had gotten a bad roll of the dice, and that it was in his genes. And he did not ever back down from this correlation of race and genetics.
He said a number of problematic things regarding race.
Right. And so he eventually lost his position at Cold Spring Harbor, which was, the love of his life, really. And so there are a couple of things that you could look at about this, but the one that caught my eye just Wednesday was an op-ed is by the person Nathan Comfort, who's working on a biography of Jim Watson. He's a science writer. And the title of that op-ed is, "He saw the true form of DNA, then it blinded him." And the subtitle is "James Watson and the Folly of Genetic Determinism." And so it's, he says, "An overweening faith that our genes make us who we are is a terrifying thing. If you think there's one single fundamental essence that makes you more than anything, you're at risk." So genetic determinism doesn't necessarily make you a racist, but it might move you to, and then he lists a bunch of sort of racist things that you might do. So anyway, I think it's worth reading that op-ed, and we can put the link to that in. And then back in 2003, Brenda Maddox, who is a biographer of Rosalind Franklin, has an article called "The Double Helix and the, quote, Wronged Heroine." And she says that Franklin's premature death, combined with misogynist treatment by the male scientific establishment, cast her as a feminist icon. This myth overshadowed her intellectual strength and independence, both as a scientist and as an individual. And so there's that. And then in 2023, Nathaniel Comfort, and who's this biographer of Jim Watson, yet to be published, and Matthew Cobb, biographer of Francis Crick. And the title is "What Watson and Crick Really Took from Franklin," and the first subheading is "Rosalind Franklin was no victim in the discovery of DNA structure. An overlooked letter and an unpublished news article, both from 1953, show that she was an equal contributor." And so the cool thing here that I learned, I don't know how I missed this in 2023, is that these two people writing these biographies went and read Rosalind Franklin's notes together. And that's how they found that, I think I put it in the show notes. It'd be faster to find. Oh yeah. So some of this wronged heroine, that she supposedly sat on the image for, this is talking about photograph 51. She sat on the image for months without realizing its significance, only for Watson to understand it at a glance. But Cobb and Comfort went through her notes together, reconstructing the development of her ideas. They say, we also found a hitherto unstudied draft news article from 1953 written in consultation with Franklin and meant for Time magazine, as well as an overlooked letter from one of Franklin's colleagues to Crick. Together, these documents suggest a different account of the discovery of the double helix. She did not fail to grasp the structure of DNA. She was an equal contributor to solving it. Getting Franklin's story right is crucial because she has become a role model for women going into science. She was up against not just the routine sexism of the day, but also more subtle forms embedded in science, some of which are still present today. And so this article from 2023 is, I think, really lays a lot of this out in more detail. And one of the things I got the biggest chuckle about is toward the end, they're writing, for example, Watson Crick had started to work on the double helix theory as a result of Wilkins's X-rays. And then conversely, she wrote, so this is about another woman, Joan Bruce, wrote, Franklin was checking the Cavendish model against her own X-rays, not always confirming the Cavendish structural theory. And then Cobb and Comfort write, it has not escaped our notice that both examples render Franklin in a position of strength, every bit a peer of Wilkins, Crick, and Watson. So they quoted that line, which you've probably heard us say on TWiV before. Alan quotes it. We all kind of quote it. You know, it has not escaped our notice that, because it's just so well known in molecular biology. Anyway, so just to let you know that there's more to the story, I will be interested in reading Brenda Maddox's biography. I've had it for a long time. I just haven't had time to read it. And then maybe this one that Nathan Comfort's writing about Jim Watson, although I'm not sure I can. But we'll see. So
I'm reading her paper, right, which is the third in the group in this issue of Nature. You know, they get the helix, they get the numbers, 3.4 angstroms for turn and all of that. But she never talks about the bases, right? She says they're on the inside, but she doesn't talk about the pairings. So I don't know. I would be interested to see what she was doing in her notes that might be part of the biography and what she was thinking about that, because that's the one thing that's different between the Watson and Crick and her article, right? How the bases are formed.
Right. And then this draft article for Time magazine never got published because it was full of mistakes, and Rosalind Franklin wanted to correct those, and then probably life got in the way, and she just was going on hell-bent for leather, working on, molecular biology of DNA. And so that never got published. So it would be interesting to see what is in that.
She also spent some of that time doing virus structure, if I remember correctly.
Yes. TMV. Right?
Yeah, I think so. That's right.
Yeah.
And so did Watson and Crick later on, too. Yeah. Interesting. All right. We did DNA. So let's do some virus structure. Anyway, an interest, obviously an important part of science history, but also the way these individuals interacted, very, very important and unfortunate in many ways. It seems to me that Crick was a reasonable person, right? He, in the sense, he didn't insult people. He wasn't a racist and so forth.
That is how the story goes.
But he said to people, "Oh yeah, we'll use this image. Sure, why not?" He didn't object to it. He didn't say, "Why don't we collaborate with Rosalind Franklin?" And they could have published it together, right? That would have been fabulous. But they wanted to do it on their own. And but as the article I quoted from, collaboration is always better.
Well, at some level they were collaborating because they submitted it all on the same day to Nature, and Nature published it all together.
Yes.
So they must have said, maybe they sent them in 3 separate envelopes, but it was all the same.
Yeah, I mean, if you look at the end, it does mention they acknowledge the contributions of Wilkins and Franklin at the very end of the Watson Crick paper.
Right.
I don't know. I wonder if they saw the versions of the other manuscripts, though, beforehand. I'm not sure they did. It'd be interesting to know. Anyway.
Yeah.
All right, classic paper in molecular biology. Now we fast forward to 2025, and and whereas this 1953 paper was with no data and with no experiments, and 1 page long, this paper is it's a huge number of experiments and many pages long.
Yes. Many different news outlets have also this paper. Sorry, I can phrase it correctly. This paper has not escaped the notice of many, many news outlets. Has been getting a lot of press, and so I was super excited. I've been super excited to read it, and it was really nice to finally get the chance to really get to dig in.
I guess we have to have somehow in the title of this episode not escaped our notice. Right? So, Kathy, you want? Oh, let me tell you the authors, and then I will.
I can do a pretty quick version of that
okay,
too. Yeah.
Go ahead.
So, well, the title of the paper is Epstein-Barr virus reprograms autoreactive B cells as antigen-presenting cells in systemic lupus erythematosus. Okay, and this is in Science Translational Medicine. I don't know if it's paywalled.
Yes, it is.
Okay, so the first author is Shady Yunus, and the last author is William Robinson. The first 14 and the last 2 authors are all from Stanford Immunology and Rheumatology. There are other Stanford departments represented, including neurology, pediatrics, surgery. Other institutions, including Palo Alto Veterans Administration Healthcare, Cincinnati VA Medical Center, UMass School of Medicine, Oklahoma Medical Research Foundation, University of Oklahoma Health Science Center, and Rockefeller University. So it's a complicated paper. They used many approaches, and as you can imagine, there are a lot of authors on this paper. And they have really exciting findings that may point to a virus affecting a human autoimmune disease. And this may be a theme that you recognize because we've been talking over a number of TWiVs about how herpes viruses in particular seem to be candidates in these kinds of diseases. So Epstein-Barr virus, which we'll abbreviate as EBV, has been making news as a candidate driver of diseases like multiple sclerosis and long COVID. It's also been linked to systemic lupus erythematosus, which gets abbreviated SLE, and I think for simplicity, we'll just call lupus, but there are other kinds of lupus, but we'll just call this one lupus for today. And so it's been known to be linked, but the why and how behind this link has not been defined. So what is SLE lupus? It's an autoimmune disease that results in inflammation and other downstream consequences that include cardiovascular disease, increased cardiovascular disease. And it's due to antibodies directed to self-nuclear antigens, and we'll talk a little more about what those are. It turns out to be 9 times more prevalent in women than men, but there's higher mortality in men. So what did they do in this paper that makes a strong link? Well, they used a new strategy in which they could identify the EBV, in other words, the virus-infected B cells from patient blood. These EBV-infected cells in blood are really rare, so this is creates a tour de force, as it were, because this is an identification/enrichment technique, and it's really important for the success of this paper. And then they did single-cell RNA sequencing from these cells, from these infected B cells, and showed that they were transcriptionally distinct from uninfected cells. In other words, they were making different messenger RNAs from what uninfected cells make. So they used additional techniques, and that's where this paper gets complicated and longer. They used these additional techniques to further characterize the properties of these infected B cells from the lupus patients. So, and then in the lupus patient samples, the EBV infected B cells had features associated with antigen presentation. And the EBV viral protein, called EBNA2, seems to be driving the activation of this antigen presentation phenotype. So the virus has a protein that seems to be driving B cells to present antigens. And these activated B cells that now have antigen presentation ability can activate autoreactive helper T cells, and that sets off a chain reaction where those T cells could then activate autoreactive B cells, including uninfected ones. And so they do some in vitro studies in B cell lines that provide support for this. So if an overall summary stated, I hope a little more simply, the data suggests that EBV infects and reprograms these autoreactive B cells, and they in turn drive the systemic autoimmune response in this form of lupus.
Yeah. So what I've known about some of this is, we have this virus EBV that I know that, Vincent and Kathy can say much more, much better things about than I can, but that partially infects B cells, one of the cells of the immune system. And there is, and leads, because it's a herpes virus, it also induces some latency, which means that there's very little transcription of certain viral genes. And there's this linkage of EBV to a lot of different disease states, and this idea that EBV is driving some autoimmunity, but the idea was sort of like, well, how is it driving the autoimmunity? Is it just sort of generally altering how B cells work in the immune system? Is it happening to infect autoreactive B cells? Is it doing other things broadly to the immune system to sort of predispose autoreactive B cells to activation, but it's not actually infecting them? Sort of how exactly is this link happening between EBV getting into B cells and B cell autoimmunity happening? And I didn't realize until I read this paper how hard it is and how rare the EBV infected B cell is, and so how hard it would be to detect that EBV infected B cell. And in some ways, as I looked at it, I was like, oh, I guess that makes sense. If it's not transcribing, how do you detect the transcript? And so a big part of this was, to me, is, that they are really the first ones who could pick out the EBV infected B cells and characterize them. Unlike others who sort of couldn't really pick out these rare cells and characterize them at this level. And there were so many times throughout the paper where I would have some sort of question that I really wanted an answer to, and then I'd have to kind of pull myself back and say, this is the first time someone's been able to actually pull out such rare cells. Some of the comparisons you want to make, you're probably asking for a comparison of like 2 cells. And so I think that one thing to keep in mind here is that this is sort of this amazing technological advance that's looking at some really, really, really rare cells, and that other people haven't been able to look at, and that some questions might be challenging to look at because of this rarity.
So the rarity is 3 to 4 Epstein-Barr virus positive B cells per million B cells in patients with lupus. That's just very hard to study, but they develop very interesting techniques in this paper that we'll talk about. But Epstein-Barr virus, of course, is a herpes virus. It is a double-stranded DNA virus, enveloped, and it infects, as everyone has said, B cells and also epithelial cells, and establishes a lifelong infection. Once you're infected, that's it. And these infected cells are mostly B cells, memory B cells, and there are different latency programs in these B cells. They're called latency 1, 2, and 3, which are mediated by different EBV genes. The virus is spread primarily through saliva. You get it from your parents or from other people in childhood, adolescence, young adulthood. It can cause mononucleosis. It can cause some cancers as well. And by adulthood, 94% of humans are infected. So they say it's one of the most common human viruses.
And that sort of makes it especially interesting to think about this as sort of, okay, if everyone or so many people have this virus, how is it that everyone doesn't have lupus if the virus is leading to lupus? And so you sort of already are getting this idea of this maybe rare event, or there's something unique happening in the lupus patient. And that's part of what this really gets at.
Now, there have been a lot of ideas of how EBV causes lupus. I'm not going to go through them all, but that's been the problem. To distinguish amongst them, it was technically very difficult. That's what they overcome here. But as Kathy has said, we already know that EBV participates in another autoimmune disease, multiple sclerosis, by inducing B cells that go into the CNS and react with self-antigens.
Yeah, and the paper that really shows that is also a 2025 paper.
Which we did, I think.
Yeah, and there was sort of in the same way as this paper had this. There's been this idea of EBV and lupus in the field for a long time. There was sort of this MS EBV idea for a long time. I know we talked about a paper in the past that did some really cool epidemiological studies on actually being able to show that. And so only now with some of these new techniques and larger studies are these things that people have kind of had an idea might be a thing. Are we actually able to sort of get this solid evidence on that?
So typically you could do single cell RNA sequencing to characterize what's happening in a certain cell type, like the EBV positive B cell, but there's so few of them. And many of the EBV RNAs, they're called EBV encoded small RNAs or EBRs. They're not polyadenylated, and that poly A sequence at the 3' end is how many of the genomics platforms, the single cell genomics platforms, work. So you can't use that for this. So they devised their own, which they call EBV-seq, and they use 23 primers specific for 21 EBV genes in a single cell RNA workflow, and they are incorporated by reverse transcription. Then they're used for PCR reactions and then sequencing. And they tested these on a EBV positive B cell line called Raji, which is a very, very well known B cell line, and they could see unique molecular identifiers of EBV. These are called UMIs, which we'll maybe mention as well, that are again mapped to the EBV genome. Then they tested it on a blood sample from an SLE patient, and they could see unique molecular identifiers, in other words, EBV genes. And whereas if you use standard single cell RNA-seq, you got nothing at all. So their technique works.
Yeah, and part of this, at least my understanding, was because of the low level of transcription of so many of these genes.
Right, but since you're now looking at so many at the same time, you get a signal, right?
Yep.
Right, enriching, yeah.
And then they combine this with other techniques, a technique called site-seq, which is cellular indexing of transcriptomes and epitopes by sequencing, BCR, B-cell receptor-seq, single-cell RNA-seq. They then sequence the genes, the EBV genes, the B-cell receptor repertoires, and the transcriptomes of blood B cells from patients with SLE, now only 11 patients, and matched healthy controls, 10. So now we're always going to be comparing the patients with SLE with healthy controls. So in the patients with SLE, they find EBV positive B cells, 25 per 10,000 sequenced B cells. And that goes from 2.3 to 82 per 10,000 in SLE. So it's a better signal than we've had before. So I guess our previous techniques have just not been sensitive enough. So that, that low estimate is probably wrong. So in healthy controls, they see 1 per 10,000 of EBV positive B cells.
And that was one of those things I kept having to go back to, is every so often I was like, well, how does this compare with X, Y, or Z in the healthy controls? And sometimes I would remind myself how few cells there were to look at in the healthy control.
Right. So essentially patients with SLE had 25-fold higher proportions of EBV positive B cells in the blood compared with healthy controls. Okay. Then they cluster these cells on the basis of differentially expressed genes and proteins. So that differentially expressed genes is given the acronym DEGS. Try not to use DEGS, but differentially expressed genes. And then proteins, which is done by site-seq. And this analysis shows you that EBV positive B cells from patients with lupus are predominantly memory cells, the CD27 positive, CD21 low memory B cell subset. Okay, so memory B cells, which is what you would expect, right? This is what we have thought before. They also want to define the latency state. Remember, there are a couple of latency programs of EBV infected cells, so they want to know. So you can define them by looking at the gene, the EBV gene expression. So they do that, and they find that these memory B cells are predominantly in latency 0 to 1 based on the expression of these Epstein-Barr RNAs.
And this is a little bit unique to the SLE patients' B cells compared to the healthy control B cells, though they don't always show all of the healthy control data. So it does look like these infected B cells in the SLE patients may have a slightly different phenotype than the infected cells from the healthy controls.
Yeah, and so they also find in the SLE patients, but not in healthy controls, a small number of B cells that express lytic reactivation gene products. So there may be a little bit of reactivation going on. We're going to get back to that later. All right. So then they want to know what is this EBV gene expression doing to host B cell gene expression. So then they measure these differentially expressed genes in SLE EBV positive versus SLE EBV negative B cells within this memory B cell.
Yeah. I found it easier as I was reading to think of this as infected versus uninfected.
Sure.
And not sort of and like try to keep the confusion down for myself.
Mm-hmm.
Yeah, with or without EBV.
Yeah.
That's fair. And so these memory B cells, they have upregulation of transcriptional program associated with very specific processes, viral processes, antigen-presenting cell function, B cell activation, and interferon-stimulated genes, as opposed to when compared to the uninfected cells, as Brianne says. They also compared the transcriptomes of EBV positive memory B cells in the SLE versus healthy control samples, and you can see that in these EBV positive memory B cells, in the SLE patients, you get upregulation of T cell activation, antigen presenting cell function, B cell activation, and interferon pathways compared with healthy controls. So you're getting a lot of gene turned on in the lupus patients, SLE patients, involved with antigen presentation and activation and so forth compared with healthy controls.
So basically the B cells in the SLE, the infected B cells in the SLE patients are more activated than the infected cells in the healthy controls.
Right. And so one of the genes that is the most upregulated gene in these infected B cells, memory B cells from the SLE patients is a protein CD70. Which is a marker of mature B cells that have been primed by antigen and can make high affinity antibodies with some T cell help, right? And they also see upregulation of genes involved in antigen processing and presentation. They also have another gene that enhances the processing of proteins so they can be loaded on MHC molecules on the cell surface. There's another protein that's essential for transporting peptides into the ER to be loaded onto MHC on the surface, a subunit of the proteasome, which generates peptides. So the idea being that these EBV-infected B cells now are better at processing and presenting autoantigens, right? The self proteins as well.
I would say antigens, but yes.
So it would be both.
We will later see them as self-antigens or autoantigens, but right now I think we just know they're really good antigen presenters.
Right.
They're very active. They're good antigen presenters.
Then this is actually a new word for me, ectonucleotidase encoding genes. Had anyone ever heard that before?
No.
I don't know quite but anyway, it is. These are genes that include ones that catalyze conversion of ATP into immunosuppressive adenosine, which is an immune modulator, which is what the virus is doing here. And so the idea is that there's some immunosuppression going on. Also, there's an increase in tumor TGF beta, transforming growth factor beta, which can inhibit T cell responses against EBV, and that would help facilitate reactivation of the virus. And so the idea is that what's happening in these infected B cells is you're getting, you're not only promoting antigen presentation pathways, but also an immunosuppressive environment that restricts T cell surveillance, and that will allow these cells to escape T cell killing and persist.
Yeah.
Right? Okay, so to additionally characterize these EBV positive B cells in lupus patients, they look at these differentially expressed genes across different B cell subsets and what pathways are represented by them. And again, they see upregulated antigen processing and presentation, T cell activation, B cell activation, et cetera, across different B cell subsets. Okay. So then they make what they call transcriptional modules. They'll have like a module of genes that are involved in antigen presenting function, a module of genes involved in B cell activation, and so forth. And then they compare these modules from in infected or uninfected cells. And again, they find similar activation across different B cell functions. For example, in the infected cells from SLE patients, they see in the unswitched memory cells, so before going from IgM to IgG, the switched memory cells, B cells of different sorts, they all have an increase in antigen presenting capacity, B cell activation, interferon module expression compared to uninfected from SLE patients or EBV positive from healthy controls. So that basically across all the B cell subsets, these EBV is activating these B cells as to be antigen presenting cells.
Yeah, so they're active. So they're very activated, and they're APCs. And while yes, other B cells in the SLE patients are also kind of activated, they're not nearly as dramatically activated as the EBV infected cells are.
All right. So next, they want to start to understand how this is happening. So they call this transcriptional dysregulation. So they looked at markers in these B cell subsets and transcription factors in both EBV infected and uninfected B cells. And they see upregulation of a variety of genes across B cell subsets. Then they want to define what one of the viral proteins is doing, this one, EBNA2, which is involved in maintenance of latency. They put all this data together with previous data on where EBNA2 is binding to chromatin. So this is called ChIP-seq. So you take DNA, which is coated with proteins, right? And if you want to know where your particular protein is binding, you break up the DNA and you immunoprecipitate with an antibody to whatever protein you want to know is binding the DNA. You purify those DNAs and you sequence them. And you can say EBNA2 binds to this particular DNA sequence. That's called ChIP-seq. So they find that EBNA2 binding is enriched at the transcription start site regions of these genes that they've just shown to be upregulated in the EBV-infected memory B cells, right? So the idea being that this is how, for example, EBNA2 is promoting activation. So in other words, we know that these pathways are activated in the EBV-infected memory B cells, and it looks like EBNA2 is the thing that's doing it by binding to the promoters of those particular genes. We okay so far? Is this good?
Mm-hmm.
They also use data which would tell them more about the configuration of DNA. So there's one of them is called coactivator ChIP-seq. So a coactivator is a protein that's necessary for transcription, but doesn't particularly bind to DNA. It does other things like modify chromatin, and also looking for, there's another kind of, there are all kinds of sequencing methods. This one's transposase accessible chromatin sequencing, or ATAC-seq, which is so cool because, some chromatin can be open and the DNA is exposed, and they ask whether a transposase, which is a DNA that can jump around and integrate, they ask where is it able to integrate by virtue of the DNA being open. So that's transposase accessible chromatin sequencing. They conclude basically that EBNA2 opens up chromatin and therefore activates transcription. And that reprograms the memory B cell to now do antigen presentation and all these things that we have talked about.
Well, in particular in the SLE patient.
In the SLE patients, yes.
That is less true if we are looking at the B cell in a non-SLE patient. So an infected B cell particularly has these features in SLE patients. The infected B cell in everybody else does not have these features as dramatically.
Right. Even if they're EBV infected.
Exactly.
Right. That's a key point there, right? Okay, so these B cells can make antibodies, right? What are they directed against? So they do more sequencing. They sequence the B cell receptor of genes of these antibodies. They find that the majority of them are class switched, so they're IgG.
So just to remind everybody what the B cell receptor is functionally, it is the antibody molecule, right?
Mm-hmm.
Yeah, so every B cell makes a specific antibody, and it's on the surface.
Sticks it on the surface
of that. So it's called a B cell receptor, but it's the antibody that it secretes eventually.
And it comes from a genetic process that involves sort of a unique sequence of a gene in each B cell, making that B cell different from the others. And so they're looking here for those unique sequences that encode these different receptors.
So then they produce some recombinant antibodies encoded by these B cell receptors that they've sequenced, and those antibodies will stain Hep2 cells, a common cell line that's used, and you can see it's staining the nucleus, which is what classically SLE patients have what we call anti-nuclear antibodies.
So I want to say again what they've just done, because I thought this was really cool.
Yeah, to me, this is the key part of the paper.
So they isolate the B cell receptor, that is the antibody, and then based on that knowledge, they can synthetically make an antibody, right? And then they can take that synthetically. Am I right still? Yeah, they can take that synthetically made antibody and show that it reacts with the nuclear proteins that are typically the ones that are reacted with in the lupus patients.
Right.
But that only happens in the infected B cells that they get from lupus patients.
Right.
And not in the infected B cells they get from the healthy controls. And so in some ways, what this is kind of suggesting, and this kind of makes a lot of things come together, is that the lupus patients, everybody at some point got Epstein-Barr virus infection, and some random B cell happened to get this virus infected or get this virus in it and have this transcriptional program. In some people, it's just, your influenza B cell.
Right.
But in some people, you're unlucky enough that it happens to be that rare B cell happens to be a self-reactive B cell, in particular, a B cell that binds to this antigen. And so what they're showing here is that lupus patients are unlucky enough to have this virus in the B cells that bind anti-nuclear antigen, or the B cells that bind nuclear antigen, while the healthy controls have this virus in some other kind of B cell.
Right. I think we get a number on what percent of self antibodies are present in people, and we're going to come to it
Yeah.
later. I don't remember what it is right now. They also do this experiment from patients with early SLE before they've been treated, and they pull out their EBV infected B cell. Well, they don't pull them out. They sequence the B cell receptor, and they make the antibodies and show that they also stain the nuclei of these cells in culture.
Yeah, and so sort of the idea is that like perhaps SLE is that you were the unlucky person who had EBV go into that B cell.
Yeah,
and
because
that self-reactive B cell instead of some other B cell.
We all have autoreactive B cells, right?
Mm-hmm.
But if you happen to get EBV in it, then it's going to turn them on and activate them. And those, as you'll see, those are going to activate other uninfected cells to also make
right. Well,
autoantibodies.
And to give a sort of expand on that and to sort of really think it through. So let's imagine, for example, that I actually don't know that I'm EBV positive, but I probably am. But let's imagine I got EBV and my B cell, the B cell that my EBV virus is in, it also responds to an Ebola antigen. That B cell has probably not really done much in my life because I have not been infected with Ebola. And so it hasn't gotten the chance to expand. It hasn't gotten any other signals. It's just sort of chilling in the background. But because we happen to have this infection of this self-antigen recognizing B cell, that B cell is now going to also be able to see its antigen very frequently and get further activation and further push towards memory. So to me, sort of, this really goes together in terms of you're having immunosuppressive effects. You might not be able to kill this B cell the way that, say, a normally activated B cell might then contract. And you're definitely seeing the antigen, whereas in another case, the B cell might go its whole life without seeing its antigen.
So since EBV infects so many people, right, could it make just randomly some people resistant to Ebola virus?
I don't know.
By having induced these
you wouldn't be able
Ebola-specific...
to do that until you had this method to look at EBV-infected B cells.
Yeah, but it might explain why there would be a rare person.
So maybe there are people who their Ebola-infected B cell is infected that you could now study with this technique, and you could see if they had a unique outcome.
It could be anyone who's not infected with a particular virus.
Right. I was just using a virus I was pretty sure I have not been infected with.
So there are always people who do not get infected.
Right.
Right? Like in a pandemic or an epidemic. And I wonder if EBV is playing some role there.
So, Brianne, I have to thank you for that, because you having explained that way really, to me, ties together how this paper is giving us a mechanism of how the EBV is.
Yeah. So it took me a little while. I was sort of confused about some parts, and then I realized that. And then looking back at some of those earlier figures about the infected B cell in the patient, the uninfected B cell in the patient, the infected B cell in the healthy control, the uninfected B cell in the healthy control, things made a lot more sense when sort of all of that linked together in my mind.
Yeah.
They then divide these EBV-infected B cells into what they call clonal families, depending on like whether they share heavy and light chain usage, CDRs, and so forth. And then they make antibodies from these individual families, from EBV infected or uninfected B cell clonal family members. And they identify families that have antibodies that stain the nuclei of cells. And they bind many different antigens. And they measure the affinity of these antibodies, and they show that the more hypermutated antibodies bind a particular protein with higher affinity compared to antibodies from the EBV-infected clonal family members. So basically, they show that these SLE EBV-infected B-cell clonal families can encode autoreactive B-cell receptors and anti-nuclear antibodies. So then they look at EBV uninfected B cells, and that have related gene usage with the EBV infected counterparts, but they have different VDJ arrangements, and they make antibodies from these cells. These are EBV uninfected family members, and they also bind nuclei. So you don't need EBV there to make these antibodies anymore. So the idea is that the EBV infected B cells are driving other B cells. They're acting as professional antigen presenting cells. They activate T cells, which then help uninfected B cells to make antibodies that are against nuclear antigens. Very sneaky.
Yes. If someone wants to know much more about this, you can look up something called epitope spreading in immune responses, because it's sort of an epitope spreading example.
Is this an accident of the virus having infected a memory B cell?
I think it's an accident of the virus having infected a self-reactive B cell that is now it lets get activated to become a memory B cell. I'm not sure the B cell was necessarily a memory B cell at first infection,
right.
But I think that it got activated frequently because it was a self-reactive B cell, and so it became a memory B cell.
I just don't know if this has any benefit for the virus, right? That's what I'm thinking.
Yeah, my question is, if in many people, the virus is infecting that Ebola B cell or that something else B cell, that foreign B cell that's perhaps never going to see anything, and so, the B cell gets a little activated in the process, but not a ton. And so maybe that a little bit of activation is beneficial to the virus. And I don't exactly know the details of it. But if that happens in just the wrong B cell, then it's the problem.
Right. All right, so then next what they did is they made antibodies from these, they have sequenced B cell receptors from 69 SLE EBV infected B cells, 15 multiple sclerosis EBV infected B cells, and 10 healthy control EBV infected B cells. And they ask, how do they bind to HEP2 cells by immunostaining? This is a beautiful figure they have here of the different staining patterns. And so of these 69 EBV positive SLE antibodies, 44 of them bind HEP2 cells in the nucleus. None of the healthy control or the MS antibodies bind either HEP2Cs or antigens that are relevant to SLE. And these antibodies will have distinct staining patterns, which is consistent with what has been seen in antibodies from SLE patients. So basically, EBV infects, as has been said, autoreactive anti-nuclear antigen B cells. So B cells that will make antibodies to nuclear proteins, and that these B cells span multiple subsets, and some can cross-bind both EBNA1 and nuclear antigens, like that we've talked about here. Okay, just a little bit more, one more experiment now. We've found so far that we have an increase in antigen-presenting cell activity within EBV-infected B cells in SLE patients, right? All those differentially expressed gene analyses express that. So they want to say, can EBV-infected B cells be antigen-presenting cells? And what they do is they use a EBV-transformed lymphoblastoid B cell line. Okay, you can take B cells and put EBV into them; they become a cell line. We call them lymphoblastoid cell lines. And they do this because the cell surface markers in the blood samples are really low and hard to measure, right? So we're using a cell line which is homogeneous. So these lymphoblastoid EBV transformed cell lines are latently infected with EBV, and they're shown before to be able to function as an antigen presenting cell. Okay, so they make these lymphoblastoid B cell lines using samples from patients with SLE who had serum antibodies against DNA in clinical tests. right? So they take white blood cells from these patients and they infect them with EBV so that they can make them lymphoblastoid cell lines. And they load these cells with chromatin complexes, right? Using an antibody to get the complexes onto the cells. And then they culture them with isolated T and B cells from the same patients, or at least the same MHC matched patients, with anti-DNA autoantibody positive SLE. So Brianna, when they're loading with chromatin, is the chromatin being taken up and processed?
Well, so the chromatin, so basically what's happening is you've got a B cell receptor on the B cell that binds to something.
Right.
That's related to the chromatin. It could be the DNA, it could be the histone, it could be anything in the chromatin. The entire kind of complex gets internalized on the B cell receptor and gets degraded. And so the DNA is not that important here. Really, it's all those proteins that are getting the histones and whatever else in the chromatin, is now able to be presented on class 2. And so now we're going to turn on, say, a helper T cell that binds to histone.
Mm-hmm.
Because we're presenting histone that was physically linked to the DNA that came into the cell.
Got it. Okay, so then they take these cells, they do single cell RNA-seq so that you can see what's expanded in them after this procedure. And they make antibodies from these B cells that they've just made, and they can identify antibodies that bind Hep2C cells. So basically showing that these B cells that they have just manipulated are autoreactive and can produce anti-nuclear antibodies. So yes, B cells can, in this case, can act as antigen-presenting cells. So their conclusion is that these EBV-infected B cells from SLE patients can serve as antigen-presenting cells, and they can activate T helper cells along with EBV-negative B cells that produce anti-nuclear antibodies.
Right. Well, and very likely some of that EBV-negative B cell activation is happening because of the T cell.
That's right.
So the T cell is getting activated. It's turning on other B cells, and it happens that those other B cells are also self-reactive B cells.
So those are the experiments. So what they show is that EBV infects autoreactive anti-nuclear antigen B cells in the peripheral blood in patients with SLE and reprograms them to be activated antigen-presenting cells. And from healthy controls, the EBV-infected B cells do not encode antibodies that react with DNA, and they don't turn on genes associated with antigen presentation. And it reprograms these B cells, and they call them access driver B cells that orchestrate activation of systemic autoimmune responses that mediate the pathogenesis of SLE. So they say it may explain why only a small number of EBV-infected people develop SLE, right? Whereas EBV is infecting the autoreactive antigen B cells, which are known to be present in the naive B cell compartments of patients with autoimmune diseases, but not healthy individuals. So,
they're present in a lot of individuals. It's just whether they're expanded.
Yeah, that makes sense. So here, this is the number I was looking for. Patients with SLE have more than 20-fold higher numbers of EBV-infected B cells compared with healthy controls, suggesting a central role for these cells.
And in theory, suggesting that something's happening to make them divide, potentially seeing antigen. So because they're seeing self-antigen, they're dividing and expanding, whereas they're not expanding in the other patients.
And they say there could be some reason why they're expanding in these patients, and that includes, defective CD8 T cell responses against EBV. EBV is doing some immune evasion. Maybe there are strains of EBV that promote B cell activation. We don't know, right? So there are lots of possibilities that could be looked at.
That sort of gets back to that list of things that they had at the beginning of of hints that they had as to what might be involved.
Yeah.
Yeah,
A long list of things.
But to me, the fact that they can actually show that the EBV infected B cell is the cell that it's the person was unlucky enough to have an anti-nuclear antigen B cell be the one that got infected with EBV.
Right.
So here's the number I was looking for. In healthy individuals, 20% of mature naive B cells emerging from the bone marrow encode antibodies which react with nuclei of Hep2Cs. And they say peripheral tolerance checkpoints typically limit the survival and activation of these autoreactive clones. So in SLE, EBV infection promotes the survival of these autoreactive B cells by overcoming these checkpoints.
Yeah. So somehow one of those 20% got hit in this situation by the virus.
Well, it's not a bad target. 20% is a lot, right?
That's true.
It's very interesting.
So Brianna, if you had to say of the 20% that emerge, but then there's these checkpoints, then we're back to
so in most people, in most healthy individuals, the idea is that all the 20% are going to get deactivated.
Okay.
And so here you had one that got infected with EBV before it got deactivated.
Right. Okay.
They also asked this interesting question. So in people who get flares of SLE, it's been associated with lytic reactivation of EBV. And so they asked the question, it's not clear whether EBV reactivation drives these flares or is triggered by them. And they go on to discuss the possibilities. They say it could be that some of the lytic expression is a consequence of inflammatory B cell differentiation rather than a driver of disease activity. So the last thing that I want to talk about, which is very interesting. So, there are anti-CD20 monoclonals like rituximab that destroy B cells. And they say that in randomized trials, so just using rituximab did not demonstrate efficacy against SLE in trials. But if you do CAR T-cell mediated therapy against CD19, this provides a good clinical benefit and long-term remission. So it's a more substantial elimination of B cells. They call it a deeper elimination of B cells than just giving patients rituximab, because that's what CAR therapy was originally designed for, to have what they call ultra deep B cell depletion of both peripheral and tissue B cells, and that seems to help with lupus patients. So that's really interesting. Getting rid of those cells, right? So that would be the key to curing the disease, I presume. Also, these EBV is also associated with other autoimmune diseases besides SLE, multiple sclerosis, of course, Sjogren's syndrome, rheumatoid arthritis, long COVID. So maybe in these cases, they say EBV infection and reprogramming plays a role in them as well. Now that we have these results, people are going to be looking at that. That's pretty cool. So we have a mechanism here and a potential therapy as well. It's neat.
I'm ready for an EBV vaccine.
EBV vaccine would be very good.
Yeah.
So someone asked on the live stream the other night, my doctor told me that it's harder to make vaccines against RNA viruses than DNA viruses. And I said, have you ever met a herpes virus? I mean, people have been working on herpes virus vaccines a long time. We've heard about it. Herpes virus, herpes simplex, cytomegalovirus, EBV. They're hard.
Yeah.
They're hard to do.
I mean, I guess the mutation rate's less, but
you don't know what the right antigen is, right? And for COVID, we got it in a year, so that's an RNA virus. So I'm not sure that was right. Anyway, it would be nice to have an EBV vaccine for sure, could prevent a lot of this, MS and SLE and other autoimmune diseases.
So are you thinking then that it would, if most of us are already infected, so you'd want to give it earlier than most of us got infected?
Yes, agreed.
Okay. Whether it could be therapeutic.
Yeah,
yet another thing.
Who knows? But first of all, I think that if we could, stop people from getting the infection,
Right.
This could be helpful.
Well, I don't know that they would prevent infection, but maybe they would lower loads enough to prevent disease,
Sure,
right?
yeah.
But, it's interesting if you were, as adults, if we're all EBV positive and we got an EBV vaccine, what would it do? Because as we saw with the HPV vaccine, it can
What I was
clear in 75% of the patients.
Yeah.
So
Well,
I think
it also becomes that question you said about lytic reactivation, and whether lytic reactivation is a problem. With the shingles vaccine, it may be that is helping with Alzheimer's disease because it's stopping some of the reactivations.
Mm-hmm.
Right.
Yeah. Good point.
Anyway, quite a paper. Very interesting. A lot of EBV activity this year. And it's all driven by new techniques, right? The new technologies. It's very cool.
So, Vincent, you gave us later this commentary. I don't know if that's open access, but you'll probably put that in the show notes too, right?
Yeah, I like that very much. That was very clearly explaining
Okay.
results. Yeah, it's worth looking. I was going to actually read it again tonight after our discussion because it's really good. Okay, let's do some picks of the week. Brienne, what do you have for us?
So I have a pick that is in some ways a pick that I have done before. However, so this pick is the virus snowflakes from the Center for Virus Research in the UK, where they have patterns for snowflakes of various viruses. However, they have updated this for 2025. So this year's is updated and has more viruses. So there are viruses. So there's a bacteriophage. I'm trying to remember. There were quite a few that I had not seen before. Cool. So I was excited about the new 2025 virus snowflakes that I will make and use as my winter decorations.
Very cool.
Kathy, is this what you gave us for one of the TWiV anniversaries? Or was it something else I'm thinking of? You gave us something we could fold, remember?
Oh yeah, those were Dutch things. I got those in Holland. They're that
just
Delft
viruses.
blue. They were kind of virus-like,
Okay.
Yeah.
Virus Schneeflocken. That's a snowflake word, huh? Schneeflocken? That's cool.
But yeah, these are really interesting ones this year.
Nice. Kathy, what do you have for us?
Well, it's kind of a pick within a pick. I have to scroll down to it. But anyway, Michigan has this thing called Saturday Morning Physics, which I hadn't gone to since before the pandemic. And actually for this one that I picked for today, I watched it online because now they're all on YouTube. And Saturday Morning Physics is for kind of the lay audience and physics people, and it's in one of those kind of steep classrooms with the big countertop in front where they can do physics experiments. And so some of the ones I've been to in the past were about music and about waves, so sound and light and various ways that physics interacts with our daily lives. And one that I remembered was about photograph 51. And a physicist explained all the parts of photograph 51 and how that tells you it's a helix and all that stuff. And so I just thought that was really cool. And of course, just like the number of times, I could hear that 5 times and I'm not sure I'd still be able to tell you unless I furiously wrote down notes. Because, why is the sky blue? I can tell you it has to do with light and wavelengths and absorption. But, so anyway, Saturday Morning Physics, this one I knew I was going to like because it has Ann Curzan as the presenter, and the title was When Physics Lost Its K and Other Language Quarks. And introduced to me the concept that physicists all pronounce Q-U-A-R-K differently from how I've always said it. And there's discussions about that. But anyway, physics used to be P-H-Y-S-I-C-K-S, I guess. Anyway, she's, Ann Curzon is a linguist. She has a show on our public radio, and now it's a podcast. Each episode is only about 5 minutes long. Mostly they take listener questions. And this one combines science and etymology. And she started off by giving us the etymology of the word Saturday, the etymology of the word morning, and the etymology of the word physics. So cool. That was really cool. And she worked it out so that she had a physics professor come and do a couple of physics demonstrations in the middle of this. It's really cool. And then she talked a lot about science writing, how we use metaphors in science language. So it got kind of philosophical. She talked about data being used as a singular versus a plural, how we tend to write in passive voice. We drop out the I when we talk. We don't say, I did this, I did that. Although I think there's more movement toward using more, less passive voice. But anyway, so, it's a YouTube. It's easily accessed. I will warn you, it's not short, but you can watch it in batches. And I think it's really worth watching all the way to the end because there's just some really fun things all the way through.
That sounds really cool.
Oh, I want to see the photograph 51 first. I want to hear.
Oh, that's a different one. And I don't know that that one's on their website because I don't know that they made, but I know who did it, so I can look for it and let you know.
Because that would probably help you with all X-ray diffraction pictures in general. Yeah. But although people don't really make those pictures, they just collect the information on a sensor. Right. But it's the same idea.
Yeah,
that's very cool.
Mm-hmm.
Well, I'm picking a book for a long time. I haven't picked a book in a long time. I haven't been reading any fiction lately, but I just started recently. I don't know why. On the train one day, I was saying, what do I have in my Kindle? And I found, The Peripheral by William Gibson, which is a 2014 science fiction book. And I have to say, I read it and I had no clue what was going on. Zero. Some vague things. And then I read the Wikipedia entry, which has got spoilers in it, so you don't want to read it. But I said, oh, so then I went back and read it again. And then I really understood it. Because the writing is a bit difficult. He will just write things and not explain it at all. It's just assuming you understand what he's writing. And then maybe 20 pages later, you'll get it.
It does escape your notice.
It does escape your notice. It certainly does. But this is about two eras in Earth. There's one in the near future for us, and then there's one 70 years after it. So the Earth is about to go through what they call a jackpot, which is essentially a horrible event which lasts many years, and 80% of the Earth's population is killed. And it's a series of things, including a pandemic or multiple pandemics, they say. And it's all because of, anthropogenic activities and so forth. And then in the future, the 70 years in the future is post-jackpot, and then you get to see what's left. And there's a statement in the book, which I love, and they say, after this jackpot and everything was a mess, then science came to the rescue. And suddenly science was used to develop carbon-sucking factories and little nanobots that could fix everything. And it's just, I love it because it's a celebration of science, even though it's science fiction. So the cool thing about this book is that individuals in these two eras can communicate with each other. It's not by time travel, but they can actually communicate in real time because in the future they have the technology to do that. And people do it as a hobby. They reach back into the past and communicate with people. And that then interferes with the evolution of wherever they've reached back to. It's called then a stub because it now is not going to follow its usual evolutionary pathway. It's going to be different because of the intervention. Anyway, there's a theme in this book, and that is someone. So the way they do this is they can contact you from the future, and then they send you to plans to 3D print a headset, which you then put on, and then you can be put into a body, a robot body in the future, and walk around and do things. So, the implications of that are very cool, and it's got a reasonably good plot. It's got good characters, and in the end, I really liked it. So, and there's also a follow-up called Agency, which is not as good. I only had to read once because then I had by then figured out what was actually going on.
I'm definitely putting this on my list.
But it's a very interesting book. Just read it slowly, because if you zip through it, which we all want to do, really read fast, you're going to miss a lot of stuff and names and what's going on and so forth. But in the end, if you can understand it, it's really very cool. I like William Gibson very much, and this is one of his more recent ones, so it's pretty cool. And there is also an Amazon adaptation, which is not good at all. They took the concept and did something completely different with it, right? So the concept that these two eras can communicate, that's fine. They used that, and they just made their own plot up because they didn't like his or whatever. I don't know. It wasn't dramatic enough, but it's not good. I watched one episode, and I was very disappointed. Okay, that'll do it for TWiV 1273. You can find the show notes at microbe.tv/TWiV. You can send comments, questions. We do have some email here, but we didn't have time to get to it, sadly. You can send them to TWiV@microbe.tv. And again, if you like our work, we'd love your support. Go to microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. That's a good thing you were here, Brianne. It would have been really hard. We would have said stupid things that would make you scream if you had listened to it. Microbe.tv on Blue Sky. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.